It's interesting. Do you ever think about that? Like what your high school self would think about all this?
Yeah, I for some reason that the concept of courage comes up to me when you mention that because I think
Really?
your progress from whatever you thought you were going to do and where you
Yeah.
get to, it depends on intelligence,
Right.
opportunity, technology, and all of that. But I think the key ingredient is courage, and it's courage to believe what you see and follow it up. Even if people tell you, who cares, or you're crazy, or whatever. And I've made a little amateur study of Nobel laureates just to see, well, what is it about these people? And what I think they all have is just courage of their conviction, and they pursued it. And I think when I think of how I progressed through science, I can identify times when I lacked courage and I didn't pursue something and I regret it. So it's really about having that faith in yourself and in what you're seeing. And when you try and train young people, the biggest challenge with training young people is that they always want to get the right answer because that's how
Mm.
academic works, right?
Right.
For a
Yeah.
lot of this, there is no right answer. What you have are the right methods or the wrong methods. But what you really need is the eyes to see and believe what you're seeing and faith in your methods. And then you make discoveries.
This is the Helix Exchange, the first podcast about the business of genomics.
Welcome to the Helix Exchange, the first podcast dedicated to the business of genomics and how it's reshaping the world across healthcare, biotech, and emerging technologies. Each episode features voices pushing the boundaries of science and business, sharing insights to help you see what's next and how to engage with the opportunities ahead. Today, we're joined by Dr. Iain Drummond, a pioneering biologist who helped establish the zebrafish as a model for studying kidney development and regeneration. His research has illuminated how organs repair themselves and how that knowledge might someday lead to breakthroughs in regenerative medicine, drug discovery, and even new business models for human health. Hello, Dr. Drummond.
Hello, very nice to be here.
It's great to have you on. We like to start with a personal story. What would you say is coded into your own DNA, scientific, personal, or otherwise, that's driven your lifelong curiosity about regeneration?
Well, I think you said it. It's really curiosity. When you look at biology and you look at the amazing complexity of how life persists and evolves, that there's almost infinite number of questions. I love solving problems and solving puzzles. And so for me, regeneration is a wonderful puzzle to solve because I think there's great fruit at the end of the puzzle.
So it sounds like you have a general curiosity about this subject, which is wonderful. Was there a moment, though, or experience that made you realize this was the question you wanted to dedicate your career to?
Well, I can trace my sort of commitment to biology back to high school with an advanced
Mm,
placement teacher I had. So
yeah.
it's maybe kind of corny, but Susan Platty trusted our little minds and let us explore problems. And so it was also, I guess, the late or mid 70s. So recombinant DNA was sort of on the horizon. So a lot of things came together to sort of present this wonderful problem of biology, a wonderful mentor who would guide your thoughts and respect your intellect, and then terrific discoveries and tools that enabled more discovery. And so I've certainly had other mentors since. My postdoc mentor, Vikas Sukatme, who got me interested in kidney in the first place, a wonderful open thinker. Coming to Mass General Hospital at a very lucky time when the zebrafish screen was just getting started. This was like the first attempt to apply forward unbiased genetics to a vertebrate system, much like had been done for fruit flies, now being applied both in Boston and in Tübingen, Germany, to this vertebrate model, with the real hope and goal that we could uncover fundamental genetics of organ formation that you couldn't study in the fly, or at least couldn't study vertebrate organs in the fly. So all those things together, I think I've been actually very lucky. I'll, I'll give you that I've been extremely fortunate to live in this time, but also have great mentors and great opportunities.
That's fantastic. I had an incredible high school biology teacher, Mr. Fisher. He is now passed away. Wow, what an incredible mentor. And he really let us lead with curiosity. And I think that makes such a big difference.
Yeah, I say to people that we, at my current institute, the Mount Desert Island Biological Lab, that we trade in science naturally, but we also
Right.
trade in inspiration because we
Oh,
have at least 20 undergraduates here in the summer who may be getting their first taste of research and just that first gee whiz moment, and they're
yeah.
they're hooked, So that was me, at one point
Right.
in my life. And you see it in these young people, and that's really what it takes because once you've discovered something,
Right.
you know, it's hard to
Ma-
go back.
Yeah, that is so true. And speaking of discovering things, you help pioneer the use of zebrafish to study how kidneys grow and repair themselves. And that gave us a window into regeneration that was almost unimaginable a generation ago. So, what's the biggest breakthrough to come out of that work so far?
Biggest breakthroughs in my work, I think, are the understanding that you can use non-human vertebrates
Mm.
to uncover fundamentally important things about people, about humans.
Right.
So when we realized, for instance, that when we were studying a zebrafish mutant, that we could inject the gene for the same protein in humans, the human gene for the same protein, and rescue a mutant and show that the human gene could function in a fish, that told us that, wow, now we can start parsing out all of the mutations that occur in humans and asking, are they important? Because that was a kind of a cloudy area about 10, 15 years ago, is how do you look at human sequence and understand whether some simple change, a single nucleotide change, was pathological or whether it was just a bystander. So that was one moment when we realized the potential of what we were doing. And a lot of the other moments have come through the ability to live image and see inside a transparent embryo an organ as it's forming and as it's going through all of the cell rearrangements, changes in gene expression. That is a unique, and really one of the main reasons I got into studying zebrafish was for the transparency of the embryos and the ability to watch cells do their thing. And the more you get into that, the more you see, and it's pretty amazing.
What was it like the first time you saw that?
Well, you sit and you stare at the microscope for quite a while. You go,
Hmm.
wow, didn't know that happened. We found, for instance, that once the kidney tubules form, they start marching as a tube towards the
Oh,
front of the fish. And so that was, we knew there was a big pile of tubule at the front of the fish, but we didn't know it formed in this kind of train wreck of this marching tube going forward. We just never thought we were going to study that. It wasn't on my horizon. But you
wow.
look at it, you see it, you can't not see it. And so we went into studying that, and I think there's still a lot of potential there to look at organ morphogenesis and cell shape changes that we just can't see if we look at fixed sliced tissues.
Right. And how does this move us closer to understanding how humans might one day repair or regrow organs?
Well, I think understanding how the cells interact and signal to each other is the key to replicating these amazing regenerative capacities of fish and axolotl and regenerative
Yeah.
vertebrates to humans. And so the ability to take that apart in a model where you can see what the cells are doing, label them with fluorescent proteins or other biosensors that report metabolic processes or cell signaling, and then see how that can be transplanted into a context where you could promote regeneration or a regenerative environment in a human. And that's the big challenge. That's the big pie in the sky, is how much we can take what we've learning from these simpler creatures or regenerative creatures and applying them to human therapies.
Just mentioned the pie in the sky. So, regenerative medicine is still young, and the gap between discovery and application, between bench and bedside, is wide. So, what's the biggest challenge in turning foundational discoveries into therapies or viable commercial platforms?
Well, that's a big one.
That's a mouthful.
But I think the promise is definitely there. So you can start with, the Yamanaka discovery that you could make human stem cells out of any cells, and that just
Right.
spawned a whole growth industry for the past 20 years in developing organs in vitro. And every organ has an organoid.
Mm.
The question is, and has been the debate, is how relevant they are to human adult organs, how translatable. In a strange way, the early organoid field reminded me of the early zebrafish field when
Oh.
you really had to fight for the argument that these are relevant to human or translatable because there's so much variation organoid to organoid. There's arguments about whether they mature fully to be really mature tissue, and there's still improvements. But I think that's the way all fields start. Nothing's perfect, and what it takes is iterative improvements, bootstrapping process, so you get to know more about what they're doing and then how to push them forward. And I think we're seeing that now in terms of reproducibility. I think we're seeing that in terms of machine printed organoid cultures where you get smaller, more uniform cell aggregates to start the process. And so you end up with a more uniform array of tissues. We're seeing that in how people are integrating stem cell derived organoids with new matrices, organ on a chip technology. And they are also highly replicated, uniform cell assemblies.
Mm.
Then you can start interrogating for drug efficacy, drug clearance, permeability properties of cells. And most along my line would be what can you do about human mutations? And,
Right.
and can you use these as models for gene therapy, for instance? And I honestly think that's going to be a big splash in the next 10 years, is both in organoids and in vivo, is new approaches to gene therapy.
When you talk about gene therapy, it makes me think about collaboration. So, what does good collaboration look like between scientists, entrepreneurs, and investors?
I think it requires each participant understanding their own strengths.
Right.
So here I am, discovery-based lab, driven by curiosity, driven by hoping to do something eventually. But really, my strength is the ability to see new phenomena, measure signaling pathways, try to model kidney regeneration or kidney repair in a fish and maybe in organoids. But I am a discovery-based person who goes deep on individual ideas. Uh, what I think we lack is the ability to take this and translate it into a larger, where you need to partner to translate it into a larger enterprise where you can test many parameters, many drugs, many gene interventions, and optimize on a higher throughput level. And academic labs are doing that. So that's sort of a gray area between this division, which I think is getting grayer and grayer as academic labs have taken on high throughput drug screens, high throughput genetic screens, and industry labs are doing somewhat similar things. I think the difference really is in their sole, or not sole focus, but intense focus on practicality and is this going to work.
Right.
Academic labs often will pursue things well beyond practicality because we're sort of driven by an idea and we really want to see. I think it's good to learn from industry that there are go, no go positions to take.
Mm-hmm.
There's an efficient way to drive towards an answer to your problem. Uh,
Right.
Start engaging with milestones, that you really want to achieve, and don't let yourself get distracted.
Right.
So that's a key strength of industry, and they're extremely focused on the end product, and on the market, which
Right.
is really driving this whole thing. So knowing our strengths, I think, would be the best way to work as a team.
Yeah, I completely agree. Now, Dr. Drummond, if you could suspend the realities of time, money, and possibility, what would you hope to do or make happen? And what would the world look like if humans could truly unlock the potential of regeneration?
Well, that's a good one. I think what you really hope for is that all of this developmental biology, all of this wonder about how organs form, and the mechanisms and signaling and the dynamics of it could all be applied to making new organs. And I think that's the evolution that's happening right now, is that developmental biologists of my generation have really completely engaged in regenerative medicine and regenerative biology, applying all of what we've learned from the last 100 years, really, in terms of how organs form, how tissues form. And so that challenge and all the stuff I have to suspend to imagine that is all of the engineering challenges. Because we know a lot of the molecules. It's really about delivering them in the right time and space and position to get things to happen. And there's a lot to be done in terms of gene delivery, um, matrices, building organs so that they're scalable. So I'm on the cells talking to each other level, and I think that's an important level to know stuff about. But the bigger question is, how do you get cells working together in a larger scale functional unit that's actually going to achieve, you know, repair for a human?
Yeah, absolutely. Now, what kind of companies or entirely new industries might emerge from that future? Again, suspending the reality, like we're talking about. What do you see?
Well, already there are large labs working in biofabrication, so that's ongoing.
Okay.
Um, there are new matrices. I think combinatorial chemistry and matrices, that's an evolving field independent of biology, but sort of partnered with it to get new ways to grow cells or new ways to keep cells happy in the long term. Huge problem is how you deliver vasculature, blood vessels to engineered organs. So that is something, how do you make blood vessel cells? How do you get them to interact with your organ so that they
Mhm.
partner for oxygen and other metabolite delivery? Um, I think in terms of new industries or new things that are popping up, I think the silver lining of the whole COVID disaster was the rapid development of mRNA vaccines. And if you just stop and think about it for a second, that technology of using lipid nanoparticles to deliver a gene is essentially a highly efficient gene therapy. And of course, that's the controversial, discussion of should I take these mRNA vaccines because they're modifying my DNA or something, which I obviously don't believe. But you could use them for that. You can use them for delivering modifying enzymes. And I think once people understand the ease of delivery of nucleic acids, and they already do understand this, but we're going to see lipid nanoparticle based gene therapies that really wipe out problems. Uh,
Wow.
I think a lot of my life has been spent trying to understand mechanisms of disease, for instance.
Yeah.
That's important biology to understand. But if you have a patient with a mutation, all they really care about is taking care of that defect in their gene. And if you
Right.
can deliver a gene, I honestly don't think people are going to care that much about the mechanism of the disease if they don't have the disease anymore. Do you know what I mean?
Yes, exactly. It makes me think of the BRCA gene. Is that a good example of
Well,
something that,
anywhere you
or,
could basically
okay.
eliminate a dominant gene by
Hmm.
disrupting it with putting a frameshift mutation in it with CRISPR,
Okay.
or supplying something to repair a deficiency. I think we're going to see a lot of that in the next 10 years.
That is very exciting. Now, every career has defining moments, successes, setbacks, and people who change your direction. What's the story from your own journey, Dr. Drummond, that taught you something essential about science or collaboration?
Well, let's see. I think, for a long time I was working in cell culture. I've done a number of model organisms, but I began to realize that you could get a lot to happen in the lab in cell culture that wasn't really necessarily what organisms use, and so I
Mm.
became kind of a purist on working in vivo. And I sort of set that as a standard for myself to show whatever I was studying was relevant in vivo and not an element of biochemistry, of cell culture on plastic. And I think things have evolved a long way away from that time. And so I think that was a major change for me when I committed to zebrafish genetics and forward genetic screening. Let nature tell you what the important stuff is. And if you're smart enough, you can figure it out. And so that I think is productive. I've seen a large shift away from fundamental genetics to what I'd call molecular biology, which is genomics and gene expression, RNA sequencing. So we're in the midst of this explosion of data in terms of just the volume of it and the multimodal aspects of it. And our challenge is to really fit that into a biological context that makes sense for disease or development or whatever the biological outcome is.
Right.
And so you see a lot of investment now in artificial intelligence and machine learning. And I think we're going to need that because our brains aren't big enough to understand what 20,000 genes are doing all at the same time.
Right.
So I think you see Genelia Farms just invested $500 million in an AI program. And what's interesting about them is they're not just investing in computers and all the AI programming, but they're also investing in new ways of seeing things. So inventing new biosensors so that you can start screening things en masse for specific pathways that may not have been visible before. So as long as you have new ways of detecting things and new ways of visualizing things and the ability to analyze it, then you're making progress. And I think that's kind of in the middle of an explosion right now. It's like a fire hose of data coming at you, and you have to be able to interpret it and make good use of it.
Yeah, well, you just mentioned AI, and the new tools like AI, single cell sequencing, and organ-on-chip models are changing how discovery happens. What's changing fastest right now in your field, and how is that reshaping the pace of discovery?
Well, I think everyone's trying to figure out how to use AI, and they're all trying to figure out how to avoid the artifacts with AI. And we're in this sort of evaluative period of what can it do for us. And there's skeptics and there's advocates. I think machine learning has clearly demonstrated its value, but the key is having a training set that's meaningful and is going to give you an answer. I think obviously it's been very powerful in pathology in terms of looking at slides and doing it much faster and maybe more efficiently than humans. I have a friend at MGH who said, well, AI is not going to put pathologists out of business, but any pathologist that doesn't use AI will go out of business.
Right.
And so it's a tool, and we should be exploiting it. The single cell explosion, we have exploited that to discover new cell types. And in the fish kidney that we were able to find in 2017, we published the first transcriptome of these zebrafish kidney stem cells. And that gave us a whole new, that's what I'm studying now. It's what I brought up here to the MDI Biolab. So that's been transformative. But again, I think in the face of this expanding massive data, we have to be continually mindful of how we're going to functionally validate it and how we're going to understand the real biological significance of it, either in disease pathways or in fundamental basic development.
Now, Dr. Drummond, how do you think about ownership and intellectual property when algorithms or models contribute to the science?
Well, just to set the context, I've always leaned heavily towards open science and shared data, and I talk too much about my work. I paid a price for that, I think. But I really believe that the thrill and the interest in science is propagated by talking about it and getting ideas from your friends. Anyway,
I agree.
that's my thing, but what I came to realize is that if you don't patent your discoveries, they will never help people because you need pharma or someone to pick them up, develop them into a therapy, and no pharma will pick up anything that doesn't have IP attached. So you need to be cognizant and aware of how to do that. And so through the last, I don't know, 10 years, I've been talking more to IP people before I publish. We try here at MDA Biolab, we try and identify any useful discovery. We just patented a small molecule drug that blocks pigment formation, a novel pathway or a novel small molecule for that. And we are mindful of any of the things we do in regenerative medicine of how they could be patented so they may be developed. I think that's the main point. AI, I haven't really thought about that very much, but you can see how this scraping the internet for data and formulating, you just hope that the patent law holds up, that the inventors can show their invention and that even if AI scrapes it up and repackages it, that you can still defend your original discoveries. Because then, you need that. I think you need to give credit where credit is due. And so maybe we'll have new ways of more accurately or completely documenting discoveries so that you can better protect your inventions.
Yeah, I think that is critically important. Now, for investors and business leaders, genomics and regenerative medicine can feel both full of promise and full of noise. Where's the signal? What deserves more attention, investment, or belief?
Well, you're right. I think there is a lot of hype. I think you have to do due diligence on looking at fundamental data and validation in either experimental models or in human tissue organoids. I think that's a value of organoids, is you can validate whether something works in humans. I think that's our big challenge with even mice. Mice have been the dominant model, but even many therapies there have not translated. So that's a real challenge in terms of hype and noise.
Yeah, it's interesting that you bring up the mice because there's so many years that we'll hear about, well, these studies on mice. And then I'm thinking, well, how does that translate to humans and how much of that is actually effective? I'm completely in favor of the research, of course, but it does get you thinking about this.
Yeah, I think we all have the same challenge. And so I'm certainly not singling out mice. The fish have that challenge. And essentially for any biotech to adopt a fundamental discovery or an assay system, the job of academia is to de-risk it for them, essentially show that this is relevant by showing it's responsive to all the same drugs that affect humans, or is it responsive to all the same genes that affect humans. And that's the burden of the work. You could have a discovery here, but spend another year or two just de-risking it so that people saw they could take it to their board, whoever's reviewing their work, and get support for it.
In your opinion, what's the next frontier where biology and business will intersect most powerfully?
Well, I think I mentioned before the advent of lipid nanoparticle delivery of nucleic acids, and that's already reshaping how we think about gene therapy. And I just see a lot of potential there.
Exciting.
The better organoid technologies will allow for more uniform screening of drugs and better detectors for pathways, biosensors that can be machine readable light output, I think is another clearly a technology it's mature, but is in iteration now to produce new tools, new ways to see things. So I'd say the ability to manipulate in vivo is an engineering challenge as much as anything right now, and that's something that can be solved and was solved for the COVID vaccine in terms of delivery of mRNA. And the ability to see new things is getting more and more mechanized. So in terms of using AI, for instance, there are these frightening experiments now where you have a machine where you've asked the AI to come up with uh, a drug screening paradigm where you might try all possible combinations of 10 drugs or something like that, or 10 pathway interventions, right? And read the outcome from maybe some kind of biosensor in cells, or maybe just cell proliferation or whatever. Gather the data, interpret the data, and reformulate the experiment to reapply a different sets of interventions or focus down on the ones that worked. And so we're going to see that. I mean,
Wow.
biology will be mechanized that way. There's a group in California that's got $1 billion or something to just integrate the robotics to do that experiment, the AI to formulate screens and plans, and then analyze and then do it. So the machines can work 24/7. You know, we're here
Right.
10 hours a day, maybe. I think. We'll see. I'll have to open the door and find out.
Let the
So
sunlight in.
I think honestly that'll change things.
Hmm.
It's like the next level beyond the 100 postdoc lab. You know, you've got the 20 machine lab doing reiterative science, uh, and it'll yield things. You know, and there are things that are amenable to that. You wonder whether can AI really think? Will it come up with a forward-looking experiment instead of basing all of its ability on what's been done before? That's the kind of question I have about AI, is whether it can it see ahead? Can it be creative?
Right.
And I'm sure I'll be shocked in the next few years about what it can do.
Yeah, and speaking of being shocked, I'm thinking about your high school self in biology and being inspired by your teacher and how incredible all of this would seem. And actually, a lot of it would probably be like, I don't even know what they're talking about because none of this a lot of this technology wasn't around back then. But it's interesting. Do you ever think about that? Like what your high school self would think about all this?
Yeah, I I know for some reason that the concept of courage comes up to me when you mention that, because I think
Really?
your progress from whatever you thought you were going to do and where you
Yeah.
get to, it depends on intelligence,
Right.
opportunity, technology, and all of that. But I think the key ingredient is courage, and it's courage to believe what you see and follow it up. Even if people tell you, you know, who cares, or you're crazy, or whatever. And I've made a little amateur study of Nobel laureates just to see, you know, well, what is it about these people? And what I think they all have is just courage of their conviction, and they pursued it. And I think when I think of how I progressed through science, I can identify times when I lacked courage and I didn't pursue something and I regret it, you know. So it's really about having that faith in yourself and in your what you're seeing. And when you try and train young people, the biggest challenge with training young people is that they always want to get the right answer, because that's how
Mm.
academic works, right?
Right.
For a
Yeah.
lot of this, there is no right answer. What you have are the right methods or the wrong methods. But what you really need is the eyes to see and believe what you're seeing, and faith in your methods, and then you make discoveries.
Right.
So
You got me curious when you were talking about there was times not having courage. Was that because it just seemed too daunting, or you thought, I don't know if this is going to work out, or are people going to, not believe in this?
I think when you have something that does work, but it's sort of
Mhm.
incremental or not that mind-boggling,
Yeah.
you can fall back on things that work and that are defensible or approvable or whatever, but not that
Right.
really earth-shattering. You
Hmm.
have to step away from these sort of comfort zones to discover things. And I think the reason I'm happy with my career progression is that there were key times when I just took the leap. And you know, moving into zebrafish, I didn't come from a zebrafish lab. You know, I wasn't trained by a zebrafish mentor. It was only when I got a job, an assistant professor position, I joined the Fishman Wolfgang Drever's genetic screen. And that was a huge moment for me, but it also meant throwing out my credibility from, DNA's hypersensitive site mapping on human oncogenes that I was working on. That's a growth. That's good angle to
Oh.
have in biology, but just to be able to launch yourself into something new does take
Mhm.
courage.
Oh, absolutely. Now, Dr. Drummond, finally, when you step back from the microscope, when you think about what you've seen in a lifetime of studying how life repairs itself, what still leaves you in awe? And what's one thing about biology, regeneration, or the human body that still makes you stop and think we've only just begun to understand this?
Well, I'm very visually oriented, and when I tend to stare at stuff for a long time, and I think looking
Hmm.
at developing organisms, just time lapses, are still
Right.
just fundamentally awesome when you try and think about all of the signaling, all the
Yeah.
cells, gene expression changes, migration, everything that's going on in an embryo. It's just how does it all coordinate, and how, if you cut it in half, does it figure out that I've been cut in half and now I have to make a new tail and a new head and recreate a fully functional organism. And I think that's the real root of life is its adaptability, and it's you can perturb it, but it adjusts. And that can be pretty frustrating when you're trying to study what a gene does and you knock it out and everything else compensates around it. But that's a fundamental property of life, and I think that is awesome.
Yeah, I think so too. Now, do you see applications for regenerative medicine beyond human health, for example, in animals, agriculture, or even environmental systems?
Well, on kind of the dark side of things, you can use
Mm,
regeneration as kind of an assay tool. I have a colleague looking at planaria regeneration as a toxicological measure of the bad stuff in the environment. So that's
mhm.
certainly one way you can use it. But in terms of regenerative medicine, uh, the pet market, the animal market, the farm market is always a lower hurdle in terms of approval of any therapies. And so I think there's a lot to be gained from uh, helping animals, but also as kind of a dry run for human interventions, where you can have a history of it working in dogs for benefit of the dog, but also see how it impacts um, potential human applications. So there are clear opportunities uh, outside of human health for regenerative medicine.
Dr. Drummond, what specifically could we try in dogs or other pets? I myself am a dog lover.
Yeah, well, the touchy thing about most pets, and especially dogs, is they
Mm,
tend to be kind of inbred, and so you end
right.
up there's very well characterized associations of genetic disease with different dog breeds. And so, gene therapies, uh, dogs maybe they already are models for interventions for gene replacement or eliminating toxic alleles. And there's a lot of money there. People, you
Yeah.
probably know, you spend a lot on your pet and you're willing
Oh,
to
yes.
go to the limit on it. So, yeah, I'd say gene
Absolutely.
therapy in dogs would be a good regenerative medicine in dogs. Um, Basically, being able to, I don't know the specific organ pathologies of different breeds or anything, but you may be able to augment kidney function in dogs and have them live longer. So I think there is opportunity there.
That would be fantastic. Could we even imagine having our pets live longer as a result of these therapies?
Oh, I definitely think so. I was just talking with a friend just now about diet and avoiding processed food in your dog
Right?
food,
Yep.
and the dogs are living longer. So there's definite opportunities there for longevity and health in the long term, I think.
That's fantastic. Could your current research be accelerated through private investment? And if so, what specific applications or goals could that help achieve?
Yes, for sure. And we maintain an open avenue for discussion with private investment. Um, so, of course, the main challenge facing everybody right now is that the landscape for future funding is unclear with the government in its current state. And I liken it to flying your airplane on instruments and landing in a fog because we don't know what our current funding situation will be. And a lot of people are really under duress. So private investment matters a lot um, in many ways. In many ways, private investment allows us to do high risk, high return experiments, which is a lot harder to get through an NIH study section. So that's one general way that private investment really helps. But specifically, we are working in my lab on engineering a plumbing problem. So basically, you can make a kidney organoid and it has a lot of the right cells, but it isn't connected to anything in terms of fluid flow. And so getting that final stage of integration or engraftment of a functional kidney requires having epithelial tubes meet and make a little lumen to conduct fluid, much like a plumbing problem. So we've figured out all the signals in there that do that in a fish because they add nephrons all the time, and that's
Right.
been our study thing. So being able to have the resources to test many ways of engineering that in human cells, organoids, in mice, that would continue our work that we've started with the NIDDK, the NIH, on this Rebuilding a Kidney Consortium, and
Right.
push that to completion. And it's a problem that really hasn't been solved. Um, and right now we have a paper out on bioarchives describing everything we've done. So we haven't patented it, so maybe that was a mistake. But I think having some investment in the engineering aspects of that is still protectable and profitable.
Oh, that's great. Now, beyond investment, what specific steps or collaborations are needed to move your discoveries from the lab into commercial applications, and how might companies use this research?
Well, like I said, I think we're at that stage of taking our basic signaling knowledge from a normal setting and engineering that, replicating it in a disease setting where we could engraft new kidney tissue. So that's where I think you need scaling up to test many different ways of doing it. Um, and I think it's not just relevant to the kidney. I think all epithelial organs have to have inputs and outputs. All secretory tissues needs blood vessels and secretory output like a salivary gland or other secretory organs. So I think that's our hope, is that our work will be useful to make stem cell derived tissue actually functionally useful. And beyond that, from a basic biological point of view, many people think that actually the final achievement of function in an organ, that there's fluid flowing through the tubes or through blood
Hmm.
vessels, it drives the final maturation. So the cells change in response to the onset of function. And so that makes them better. And it
Right.
may be the final step that's missing in a lot of these in vitro tissue-based organs. So that's the other thing we have our eyes open for, and I think I'm hopeful we'll have an impact on.
All right. Well, finally, what unconventional advice might you give to someone looking to get involved in this space?
Keep your eyes open,
Okay.
stay courageous,
Mm.
and when I say keep your eyes open, I mean there are so many technologies that can be applied that maybe no one's tried, in terms of looking for new ways to do things, speed things up. Talk to your friends working in robotics. Talk to your friends
Right.
who are computer scientists, and really engage in a cross-disciplinary investigation, because I think that's really where the excitement and the real innovation happens.
Right. Courage and collaboration. I think this has been such an incredible discussion. Dr. Drummond, thank you so much for joining us on the Helix Exchange. Your work reminds us that the business of genomics isn't just about data or markets, it's about reimagining what's possible when we understand life at its most elemental level. And for our listeners, whether you're in finance, biotech, or the business of building what comes next, the question is how you'll engage with this new biology as it moves from science to system and from insight to impact. And if you're ready to stay ahead of the curve and explore the opportunities shaping tomorrow, make sure to follow, subscribe, and share.